We have seen in previous lectures that it's possible to formulate simple mathematical models that allow us to understand a repertoire of interesting, both quantitative and qualitative phenomena. The models I've been working on so far are models that deal with continuous variables. So we have a population of organisms or molecules that change in time following differential equations, right? Equations that gives us a functional form for the speed of change in the variables that are relevant. For example, if you think inside cells the way that transcription and translation happens, it's so far our approximation was there's a continuous production of molecules and degradation, right? And that can be written down with simple kinds of equations. There's plenty of things that we haven't considered here, right? For example, one that is especially important is these linear chains, right? It's just a little part of the whole story. Now, imagine that, for example, this protein here, right, is a transcription factor. In other words, a protein that is able to interact with DNA and perform a regulatory function. That means that this protein could actually influence the activity of its own gene, all right? So how do we model that? The other thing is that in a much more general framework is now if I want to go into the scale of the cell and think about the processes that happen inside the cell, I need to ask myself, what is the right metaphor? Is a cell something like a computer? This is an analogy that is used very frequently. And what could be the analogy here, actually? Well, I can think in the cell, in a simplified picture, as a system able to gather information from the environment. So I could have, for example, sensors, molecules that are in a way located on the membrane that can capture information from the environment, but could also sense information within the cell, right? Sensing systems that in a way evaluate the inside state of my system. Then these sensors might send signals to some cascades, right? I just write down these, some intermediate steps here. And these somehow, and I use the CPU term in a kind of loose way, are integrated by the machinery, right, that makes decisions. These decisions might actually involve feedbacks, right, where the sensing, it's also the sensing of my own state, right, but also from the state of the environment. And then all this integration, right, might affect particular outputs in the system, right? For example, the movement of flagella, right? So the movement of the bacteria as they, for example, search for nutrients, right? In the old domain of cybernetics, that will be the picture that I have sensor units, I have processing units, right, that usually make comparisons, and actuators, all right? So parts of the system that allow to actually perform functionalities, all right? Sensors, processors, comparators, and actuators. Is that all? Is that the way we can think about a cell? Well, that's not very different from a machine, right? And now the question that I want you to think about, right, and I propose you to try to make an essay on that, is let's compare a cell and a computer, right? Remember some things. Inside cells, inside the computer, you will have a number of units, a big number of units, as well in a cell, right, that have fixed positions, right? And there's an architecture that has been built, it's called the von Neumann architecture, has been built to store and manage information. Within cells, this simplified picture is really far away from what we see in reality. It's a fluid, crowded environment where molecules are just being created and destroyed, moving around in an extremely noisy and crowded scenario. So, is this something that fundamentally changes the analogy with the computer? Think about it. What kind of list of properties can you make? All right? All right. And now there's another important message that brings the analogy with the computer. In computers, we use a discretization of information, right? Zeros and ones, right? Everything that you can actually define in terms of a signal, a message, whatever you want to think about, can be mapped into a sequence of zeros and ones, a discrete sequence. This is what we call the Boolean logic, right? From George Boole, the mathematician who actually brought the idea of Boolean logic as a formalization of the philosophical domain of logic, right? In zeros and ones. Is this something that we can also use in the biological context? Well, think about the following. When we analyze these problems in the continuum, right, we often describe whatever is the variable, right, dynamically, in terms of something that describes some trajectory in time. This is called a time series, and goes into some attractor, right? Sometimes we find that there are several attractors, right? Some alternative states. But if we ignore this, ignore the transient, sometimes the transient is very important, sometimes it's not so important, because maybe when you look at the real world, what you see is this, right? Everything that had to happen already happened, right? And you are in the fixed point. Think, for example, in this problem here, right? Imagine that I look at that in a more qualitative way. For example, I have a gene. I'm making a very simplified picture of genes here, right? Consider the promoter where the DNA polymerase binds, but also where other things happen, right? All the transcription factors play roles, etc., etc. Imagine that I have my polymerase, okay? And you know that if the polymerase binds this, right, there's transcription, right? So I could have, consider two possibilities, right? In one case, the polymerase binds my promoter, and what's going to happen is that it will read the sequence of the gene, and I'll have an RNA, and so on and so forth, okay? Now imagine that it is a repressor molecule. A repressor molecule, a transcription factor, that if it binds, right? If it binds here, do not allow the polymerase of performing its function. There's no transcription, there's no RNA, right? So this step here doesn't happen, okay? All right, I'm describing this in a discrete manner, right? If the repressor is present, there's no product. If the repressor is absent, right, I have a product, right? I can write down this using a logic table, which is the following. Repressor, RNA, right? If it's not present, use a 0, RNA is present. And in terms of logic, I write down this as a 1, and the other way around. This is what is called a logic table, okay? Or truth table in formal logic. And this particular table represents what in electronic circuits we label as a NOT gate or a NOT table, right? The mRNA is the negation of R, right? When you have a 0, you have a 1, 1, 0. All right, so no differential equations, but I say something about the dynamics of the system. Qualitatively, I say exactly what happens when one particular element is present or absent. What's the impact on what is going to be observed? Not interested now in the amounts, right? But I just go into the presence or absence, right? So going to the Boolean logic, right? So is biology Boolean? It turns out that to a large extent it is, right? Because, for example, when you look at differentiated cells, cells in your organism, neurons, hepatocytes, whatever you want, you have stable cell states, right? Where some genes are active all the time, some genes are inactive all the time, and what you actually have is a series of on and off genes that is stable, right? And that allows this particularly discrete description in the genome to map into something that is a stable cell type, right? And you again can think in terms of a discrete collection of states. Is this a complete picture? Well, we'll say the analog computation, the continuous computation is also there. But the Boolean description seems to be pretty fit, pretty good, right? In particular, and you will see that in a seminar, right? Because the way the things respond to inputs, the input-output logic, it's very often highly nonlinear. Let me just make a little abstraction here without proving it. We will, right? The idea is that if I have, for example, something that is under some concentration, some signal, right? It's going to be a product that could be, for example, a concentration of a given signal that comes from a transcription factor or a cofactor, whatever it is, right? Something that can affect the output. And P, the product, could be, for example, the concentration of mRNA, right? But it's much more general, right? And for example, one possible way of finding a response, you can see something like that. This is very common, extremely common. And the reason of that comes from fundamental nonlinear properties of these systems. Very low concentration of this, right? I have essentially no response, which makes sense, right? Intuitively, if something has to trigger a response, but the concentration is extremely low, you might not get there, right? Whereas there's a saturation at high concentrations, okay? And the reason of that also is that once you cross some thresholds, you put more molecules there. But for example, if there's a limited number of places where you can actually act, nothing else is going to happen. You are saturated. And that allows actually to think in an approximation that is the approximation we're going to use, right? That this essentially is a 1-0 threshold kind of function, right? That if response is so nonlinear, I can think in terms of a Boolean logic, right? Let me make some parentheses here to comment about something that I think is important to mention, one consequence of that, in terms of critical thinking, right? If you think, for example, it's called a response function, right, in engineering. If this, for example, is a drug, concentration of a given drug or a vitamin or something that you take, and this is the physiological effect, right? That's a typical scenario. You also have a nonlinear response. And that's have an important meaning, and keep that in mind, right? Is that if you take vitamins, for example, and you take a lot of them, so you're here, okay? What's the outcome of that? Well, somebody said your P will be very expensive, right? This makes nonsense to take so much. It's not going to improve anything else. Whereas here, right, at almost zero concentrations, right, the effects are zero, right? It's no effect. I will put here homeopathy, right? The famous idea that if you dilute in concentration something in such a way that essentially you have no molecules there, there's no effect, right? There's just magic. Or Reiki, okay? If I put my hands on top of you without doing anything, right? That has no effect either, right? Let's have that in mind. We are scientists. Okay, so now the thing is, how can I formulate in a Boolean fashion, right, the repertoire of possibilities that allow me to describe functional changes in my system. To do that, right, we'll start to first describe the set of logic gates that can be actually defined, right? And then we will go into the genetic logic. How can we implement them using genetics? And that's important for us also, because actually we can engineer this logic in cells, right, using synthetic biology. For example, first thing that we are going to do, let's consider a circuit that we have already seen, okay? We saw it before when we started to explore reliability theory. We have a circuit with two switches, okay, and an output. An output can be a light bulb, right? If the light bulb is, you see the light is a 1, you don't, it's a 0, okay? And my switches will also have assigned values. 1 if it's closed, 0 if it's not, right? Open 0, close 1, okay? Look, I can build a logic table for this system, okay? Because I have two input states, right? This can be two possible things that they can use to actually trigger responses in my output, right? How many possibilities are there here, right? 2 to the 2. And I'm going to order them in this way, right? Y 0 0 0 1 1 0 1 1. This is the way of ordering in binary description, right? From 0 to 3, right? Right? My numbers in decimal will be 0, 1, 2, 3. That's a description in binary digits, right? So what happens when both switches are open? Well, clearly there's no output, okay? What happens if one of the switches is open? Doesn't matter which one, no output, right? The only possibility when I have a light bulb giving light is when both are closed, right? That's your first example of a Boolean gate, right? This is called the AND gate, right? The AND gate, sometimes we write this here like this as AND, S1 and S2. It's a form of saying logically this, or you will find out also this notation, okay? Requires that both inputs are 1s, otherwise the output is 0. Very good. We are not going to represent the AND gate in this way. We're going to represent it using a particular drawing where here I put the inputs, here I put the output. This is a logic representation of the AND gate, right? For this table, I represent the gate in this way. This actually could be generalized, right? And it's important to have that in mind also. What happens if I have not 2 switches but n switches, right? It's very easy to see, right? For n switches, right? That the gate will be essentially the same thing because, right? Because only the output will be using that notation, right? S1 and S2 and Sn, okay? Or if you want to make it in a compact form, this is the N AND gate. You want and, all right? So this is an example of a logic gate. If I ask you to define a genetic circuit that implements that gate, right? And again, as I was saying, nowadays we can actually decide to make this inside the cell and use for that genetic engineering with genes and promoters and whatever we need coming from different species, right? So we can actually put inside an E. coli cell machinery that is from a phage or another kind of virus or a human gene. We can do, in principle, a lot of things, right? What kind of genetic circuit represents a Nand gate, right? There's a number of examples, and part of the exercises are going to be about how to build it. Well, let me take an example. Suppose I have this gene here that synthesizes, well, gives this as a gene product, this protein P1. I'm not writing the intermediate steps here, right? Whereas another gene synthesizes or gives rise to another protein, P2, right? And I have a third gene here. Okay, I was describing this the direction for transcription. Gene 3, okay, which can also give a protein. Typically, we will use the idea that is common in synthetic biology that the final gene that in a way gives the signal, which is the output of my system, is going to be, for example, a fluorescent protein, right, like GFP, right, green fluorescent protein, something that I use to actually know if something is happening or not. Have in mind that that doesn't mean that there's no functionality. I could have another gene here that actually performs a given function, right? My GFP just says it's on or off. And how can I implement my AND gate here? For example, imagine that this protein and this protein, right, form a complex, what's called a heterodimer. One important thing, dimers, where the same kind of protein or different, are extremely important to actually allow us to think in terms of Boolean logic, okay? And that this heterodimer is required, and I'm going to put this here, right, with a positive sign, is required for this gene to transcribe, right? What is the meaning of that? Well, that for example, I have this gene, imagine, right? My polymerase, right, is because the kind of promoter I have may have a hard time to actually bind to it, right? It might do sometimes, or maybe most of the time it doesn't. It cannot attach there. But in the presence, oops, of a given molecule, let's say an activator, right, that helps the polymerase to actually bind and transcribe. So this means that we need them. And what happens here is that if the heterodimer is present, right, I transcribe the gene. And look, this is very easy to see that if I put now my table for protein 1, protein 2, and that could be RNA or GFP, right? If I look at whether or not, in this case, what I'm saying is the genes are expressed or not, right? But you see, you will have a heterodimer, the molecule that you need to actually perform this function, if and only if both genes are being expressed, right? If not, I will not. So no, 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 yes, right? This is our first genetic gate, the AND gate. And this is one example, and we will look for the other examples, right, of actually one collection of possibilities, right? Actually, we are considering functions. My functions here are defined by this table. My functions now are discrete, right? And for this particular kind of system, right, where I am working in binary, so my state of variables, let me write it this way, is 0, 1, Okay? This is 2 input, 1 output functions. So this is an example of one of the possibilities, right, of mapping 2 bits, zeros and ones, into 1 bit. All right? Let's go for the second example. So we have seen so far 2 examples of logic gates. One are already explicitly defined, the AND gate, when only this combination of S1 and S2, right, both ones, is there. And remember, we use this notation, S1 and S2 as a way of defining it. And we didn't define the logic gate, but we already mentioned this negation process where the presence of a repressor that does not allow polymerase to bind is actually something that defines, right, a negation. If the repressor is not present, the polymerase can bind and I have expression, right? Whereas if it's present, I do not have expression, right? This is also a logic gate, right? This is a 1 input, 1 output, right? So in that case, could be an example of a function that goes from sigma to sigma, right? It's a small subset, as you can see. And the representation is this one here, right? This is for the negation, okay? And a way of representing that, it would say that mRNA is the negation of R. Of course, mRNA and R now are present or absent. We are playing the Boolean domain, all right? Okay, so we have 2 examples, 2 genetic gates. As an exercise, think about how would you build a NAND gate with a different mechanisms from the one I just proposed, right? And now that you know that inhibition is a one way of doing that, that can give you some insights about how to do it. What's the next gate that I want to define? It's again something that we have seen already. Again, we're looking at reliability, right? When you have a circuit with 2 parallel switches, right? Let's do this again. I have my light bulb here, okay? Now my switches S1 and S2 behave very differently in terms of their impact on the output. If one of them is closed, so if one of them is 1, right, I have a flow here and I have an output 1. If the only case against that is that both are open, right? So it's very easy to see that if I want to build my logic gate, again with the set of possibilities here, right, organized in this way, the only state where I have a 0, right, kind of the opposite of the previous case, is when both are open. If not, I have this, right? So that's the OR gate. We'll describe this using this symbol, right? S1, S2 output. This is the OR gate, which often it's indicated as S1 or S2, okay? Well, that's something that's a gate that doesn't require as much constraints as the AND gate. And one way of implementing it using a genetic circuit is simply to consider, again, let's take our example before. I have one protein here from one gene. I have another protein from a second gene. Oops, excuse me. All right? And these 2 proteins are the input of a third gene, right? Okay, not assuming anything about how each one of these is being expressed. And let's assume that they are both activators, right? They are both proteins that make possible for the polymerase to actually perform transcription, right? So this is enough for doing that. This is enough for doing that, right? Clearly, if any of them is present, I will have expression. I have my GFP. This, the name usually that we use for this is a reporter, right? A protein that tells me whether or not it's an output signal that is 1, okay? Could be green, red, whatever, okay? And you can clearly see if either P1 or P2 are present, I'm going to activate the gene, right? So I'm exactly in this scenario here, all right? Very good. As we, again, as an exercise for you, think about an alternative design circuit, right? We can combine also inhibitions that actually implements this, all right? And just for the record, in case we need it, if I had multiple inputs, right, I could define again an input OR gate. Well, an N OR gate, right? The logic is exactly the same, right? And it's exactly the same as we also use in reliability theory for the multiple switch parallel circuit, all right? Very good. Before we proceed to going into more gates, okay, let me point to something that is really important, and you will see that the consequences for that in our understanding of the computational logic and power of biological systems is really important. This set of functions and or not, right, is known as a functionally complete set. Meaning what? Meaning that using these gates, right, I can build any Boolean logic circuit I can imagine, right? So that is going to mean jumping into whatever kind of circuit from multiple inputs and outputs, right? Said in another way, we could actually build any computer, right, provided that this is all we need, right, and at least in terms of the logic, by using just the elements of this set, all right? We'll go back into that in a moment using one particular kind of gates that allow to make this still more specific, okay? And so far we have this, right? Before I go into these final two gates, just want to mention that this here, right, the output of my table tell us immediately that there's a number of possible tables that you can actually build, right? How many? 2 to the third, right? So there's a number of combinations that you can use. In this case, it's 8 possible logic tables. But important to point out, this is a combinatorial thing. As soon as I go into 3, 4, etc., this rapidly increases. And something to have in mind, because of course inside cells, these are just small modules maybe that I can define with a few genes. But within a cell, we have thousands of genes that can be interacting, right? What can be the outcome of that? And what's the potential for this? All right, so we're not going to derive all the possible examples here, right? But for as exercises, you will explore some of them. There are 2 gates that is important to define because they have relevance for many reasons. Well, one is the NOR gate and the other is the NAND gate. Let's start with the NAND gate. The NAND gate is obtained, right, by combining two gates, sorry. One is a NAND gate, right? The second is a NOT gate. All right? So this is your first combinatorial circuit. This is the AND gate, all right, that we know, and this is the NOT gate. And first of all, it gives you the first insight into the power of combinatorial logic, right? Because we can actually perform as infinite computations that you can imagine, as infinite combined logics you can imagine. And this is very easy to find out how it works. S1, S2, right? S1 and S2, right? Let me make the steps here. We will not do that typically because it's quite intuitive. What happened with this, right? This is this here, the output here, right? Is the output of the NAND gate, sorry, of the AND gate, right? So only one here, okay? Now this is the input of the NOT gate. So the output here will negate whatever it comes from here, which means that it's just inverting everything that comes from here. And the way we represent in terms of the formal logic, the negation of this is with a horizontal bar saying negation of something, all right? You might have seen this with another kind of formalism. Sometimes we use this to indicate negation, or we could say negation of S1 and S2, but there's such as a choice, right? So that's the NAND gate. That's the NAND, the logic of the NAND gate, okay? Right, we don't represent the NAND gate in this way. We want to make it some more compact, and you can see that from the notation of the NOT gate, we can actually represent that in a nice way, like this. All right? This was 1, 2, output. This indicates that it's negation, not the NAND gate. Why the NAND gate is important, right? I could have combined other things. Well, the NAND gate, this gate here, is important because it turns out that this gate and only this gate, right, and the same is going to happen with the NOR gate, which is the negation of the OR, right, can be shown to be able to generate any other gate, right? For example, imagine I want to generate from this and only from this a NOT gate. Sounds like a puzzle, isn't it? Because this is 2 input, 1 output, right? How can I do that? Because the NOT gate is 1 input, 1 output, right? So we can do it the following way. I'm going to actually exploit my NAND gate, right? But now with a little trick. I can convert my 2 inputs into only 1, right? So to get my input output this way, okay? So this input bifurcates exactly in the same way, in such a way that if I have a 0 here, I will have 2 zeros here, right? And I can know exactly what it is this. Input, output. If this is a 0, I send 2 zeros to my NAND gate, right? But 2 zeros in the NAND gate means a 1, right? Whereas if I send a 1, here is 1, 1, right? And the NAND gate gives for 1, 1 a 0. So I have a NOT gate, right? The same is possible, for example, for implementing an AND gate, right? How can I do it? Well, repeating this scheme, right, twice, right? So again, no, excuse me. I want an AND gate, right? What I'm going to do is repeating this scheme here. Like this. So actually I'm using two NAND gates, one here and one here. Right? So here. And using this combination, I give you this as an exercise, very easy. Input 1, input 2, output. You have to do the same logic, right? Now you have to take the table here, look at what happens when you go into the NAND gate and you have the same outputs here, right? And you can show that this is nothing but a NAND gate. It can be cumbersome sometimes because, for example, to get into an OR gate, you need a bit more complex circuit with 3 parts, right? But the main lesson is it's possible systematically to find out, right, how to combine just one type of gate to obtain the whole repertoire. All right? Try to do that with the OR gate. Okay? You can find in the literature and the internet the whole set of examples. And that means that if that set was a complete functional set, right, this gate is in itself a complete set, right? It can generate any circuit that you can imagine, right? That's one of the reasons why, for those of you familiar with circuit design, you have these little chips where you have several NAND gates working there, right? Well, the same can be done with the OR and NOT. Before we propose that for you as an exercise, let me actually elaborate into the genetic circuit here. How can you build a genetic circuit that implements a NAND gate, right? And an easy trick here, right, is to think in the way I have generated a combinatorial expression for the NAND, right? A NAND gate and a negation, okay? It should be easy to do because actually we have the AND gate and we have seen a genetic circuit for that. We have a NOT and we have an intuition of how it works with a repressor, right? And as you can easily imagine, one way of building a NAND gate is to take two elements, two proteins that form a dimer, right? But they act not by activating, but by repressing. So we need both, right? And then the heterodimer or the dimer, right, represses the gene. You can show that this is exactly what implements a NAND gate, right? You write down the circuit and think about another possibility. Think in another circuit that is not using that kind of particular configuration, okay? So my last gate that I can define, right, and you will build it completely, is the NOR gate. The NOR gate, that's not going to give us more insight than what I've just said, right? The NOR gate is, as you can imagine, is a combination of OR and NOT. It's a negation, right? It's indicated in this way. You can see that I take the OR symbol at this little circle, right? And of course, since it's the negation of the OR gate, you take the table of the OR, right? And you invert everything. You'll see that this is what happens, right? So I propose you as an exercise here too, right? Look at this design. It's possible to show exactly the same because NAND and NOR gates, right, are universal in the sense that they can generate any circuit from there. And you can actually do the same thing. You can try, right, how you do a NOT gate from a NOR gate, for example, right, or an AND gate from this, okay? And I'm asking you to find out a genetic circuit, right? How would you engineer a genetic circuit so that it implements a NOR gate, right? The importance of these gates does not end up here. It's much more general. And in particular, when we will move into neural circuits, right, where again a formalism can be used to actually define how neurons behave as Boolean objects, that will have important consequences about what can we say about the computational power of brains.